Thank you very much, Marco, and pleasure to be here. And thank you for coming. I know that some of you might have been up late after Italy's victory last night. So I'll, of course, talk in English, mainly for the simple reason that your English is an order of magnitude better than my Italian. So the work I'll talk about is work that essentially started with this, one of my brightest students, Andrea, who came funded by the Royal Society. He worked with me for 2 years, and this is when we basically started this work. And I should really sort of acknowledge him because he was a really superb student, and he's back in Naples now. Okay, so the work I'll talk about, let's just briefly sort of give you the roadmap. And I'll go into some details in some topics, and I will not go into some details in some topics because of time. I hope that I've chosen those, wisely. I don't know, to be honest. So I'll briefly introduce what is this I'll talk about in terms of the cell mechanics. It's the cytoskeleton, but I'll specifically focus on one aspect of the cytoskeleton, which are these stress fibers, and the mechanical influences is what I'll discuss. Then I'll go into what we would traditionally think about as, from a materials perspective. We'll talk about what you could call a constitutive model for this element of the cytoskeleton, which is these stress fibers. And I'm calling it deterministic because it's going to be a constitutive model. It'll give me some energy functional or something like that at the end of the day. But then I'll go into what I would say is the main focus of this talk, which is saying that this deterministic business, thinking about this as a constitutive model, is essentially, in my opinion, sort of rather simplistic from a biological perspective, mainly because you never see sort of something which is deterministic in a biological system. So I'll then go on to talking about what I will call statistical mechanics of living cells. Now, these are significantly larger than proteins or molecules, so why should statistical mechanics apply? So we'll discuss that. And these would be the 3 topics that I will basically cover in that, which is the key aspects being separation of timescales and the entropies that would be occurring at the different timescales in that. Okay, so starting off with what is the cytoskeleton, most of you might know this, but just one quick slide. So if you think of a eukaryotic cell, that is a cell with a nucleus in it, most cells inside our body have nucleuses inside it, other than red blood cells, for example, will not have a nucleus, so those will be excluded from this discussion. Have a cytoskeleton which comprises of lots of fibrous elements, and they comprise of proteins called actin, microtubules, intermediate filaments, etc. And these cytoskeletal elements, this sort of skeleton, as you could think, is what provides the structure for a cell. So for example, these animal cells basically have a cell membrane, which is extremely flexible. They don't have a cell wall like in plant cells. And so what is providing the structure for these cells are these sort of fibrous membranes, fibrous skeleton that exists inside there. So for example, this particular thing, which is a sketch of what we would call a stress fiber, it comprises of 2 proteins mainly, called actin and myosin, and we'll talk about this one particular element a lot. And the main differentiating feature about thinking about this as a skeleton is that this is not some static scaffold. It is extraordinarily dynamic, as we will see as a moment. It's constantly evolving with respect to, we don't know exactly what, but maybe external stimuli and other things. So that's the sort of, this skeletal structure of these fibrous elements is what I'll be focusing about today. So let's say, what are these stress fibers? This is one element of the cytoskeleton that I will focus on, and we'll not, for simplicity, we will not take into account all sorts of other elements. So if you take a cell and you stain it for actin, which is one of these proteins, actin is just a 1 nanometer ball, you can think of it that way, and you stain it for that, you see that's what is stained in green. You see these fibers here. These fibers are called stress fibers, and they end in some things called focal adhesions. Focal adhesions are where these stress fibers anchor to the substrate, basically. Okay, so what do these stress fibers consist of? They consist of this, as I said to you, this 1 nanometer ball, which is this actin. And these actins will then, in normal state, they're sitting inside a soup, inside the cell. They can sort of polymerize, and they will form these long chains. Then these long chains are what I've tried to draw here with these red bits. And then there's other protein, myosin, which is one of the most important proteins in these stress fibers. They will also sort of they will get together and form these bipolar filaments, and then they will go between these actin chains. Now, stress fibers is something that we would know about in normal circumstances extremely well. This business, my muscle contracting, is because they consist of these, what you could call muscle fibers, but muscle fibers are these actin, myosin sort of filaments together. And these myosin are motor proteins, basically, and these motor proteins will start to sort of do actions like that, and then they will result in contraction, basically. So they're called stress fibers because they actually will generate contractile stresses or contractile velocities, and that's why we would call them stress fibers. They will generate, they're force-generating agencies. So this is the one element of the cytoskeleton that we'll focus on, and you'll see why is it that I've chosen this one element for this talk as we go along. Okay, so that's the general thing that we'll talk about. So what is it that we want to explain, and why is it that I feel that mechanics can make a big contribution to this field? So let's just think of very simple observations. So if you think of putting cells on a glass substrate, so this is a glass substrate, and you've just put lots of cells on it, and you've stained it for actin, and you've stained it for the nucleus being in blue. What the first thing that you notice is that, and then you've stained it and you've frozen the system in that way, is that no two cell hairs are identical, basically. Every cell that you're seeing here is somewhat different. And then if you take one cell and you just observe this over, this is a movie over approximately 10 hours. So this time lapse here is in hours. What you're seeing is that this cell is fluctuating, basically. It's not a static agency. So the claim that I'm going to be making is that all these different shapes that you're seeing here is that you're catching every cell in this ensemble at different states of this fluctuation, basically. And that's why you're seeing all these observations. But this fluctuation, so I call it a fluctuating equilibrium. We'll discuss what we mean by that fluctuating equilibrium as we go along. So this fluctuations is critical to life, basically. You'll see in the biological literature, people will say, oh, cells detect the environment, they feel around, and they sort of decide to move places. How is it that they actually manage to detect the environment? It's these fluctuations. This is the mechanism by which they start detecting the environment. And so really what I feel what is critical is to be able to predict and understand these fluctuations, to understand sort of what we would call the response of cells to different kinds of environments, basically. So what are these responses that are very interesting So there are various cues, as the biologists would call it. So let's think of mechanical cues. These are experiments by Dennis Disher's group, old experiments, really. So if you put cells on different stiffness substrates, this is a 1 kilopascal gel, and this is a glass substrate. So what you see is that on the 1 kilopascal gel, the cell is rather contracted, while on the glass substrate, it's spread enormously. And this is captured in this plot here. So if you just look at one collagen density and you go upwards, this is the mean area of the cell on a soft substrate, but as you start increasing the stiffness of the substrate, the cell area is increasing. What's even more interesting to me is the following, is that in biological work, you always put massive error bars or standard errors, as you would call it. So the standard error on an experiment done on a soft substrate is significantly smaller than the standard error that you're seeing on a glass substrate. Now, if you had said to yourself that this is inherent biological variability, that every cell is different, why is it that you are getting a much bigger variation on a glass substrate versus on the soft substrate? So there's something there for us to understand, basically. But what's even more exciting from a mechanical perspective is that you take these cells, this is even older experiments done in the 1980s. So these are control experiments, 45 minutes, 90 minutes. You come and add a reagent called BDM. BDM, basically, there are lots of these reagents that I'll be discussing. If you add it, it kills the myosin activity inside the cell. So it basically stops the contractile behavior of the stress fiber. It's stopping this from happening. Now, you'd say to yourself that if you stop the contraction from happening, the cells would want to spread more. That is the sort of standard thing that you'd expect. But look what's happened. In the control experiment where there was no BDM added, the cells spread a lot. But when you added BDM, you killed the contractile activity, the cells completely became sort of globular, basically. So they did the opposite to what you would expect, basically, that the contraction, for some reason, is actually resulting in the spreading of the cells. This is sort of counterintuitive in some ways. Another mechanical cue is that if you again it also affects the shape of the cells. So if you put cells on a glass substrate, they take these very sort of elongated shapes, but on a soft substrate, they become more circular in shape, basically. And this is captured in the cell aspect ratio. Here is that you can see that cells on glass substrates can have very large aspect ratios, but nearly no cells on soft substrates have these very large aspect ratios. And again, I keep emphasizing that whenever you see any experiments being done, you see always these kinds of box plots, error plots. You never will see a deterministic sort of plot, basically. Another mechanical cue is that if you take stem cells on different stiffness substrates, these are again Dennis Disher's experiments, is if you put them on, these are stem cells, if you put them on very stiff substrates, you basically see that these cells have differentiated into osteoblasts, again with a probability distribution. Over this range, they differentiate into osteoblasts being bone cells. On slightly softer substrates, they're differentiating into myoblasts, which are muscle cells, and then even softer substrates, they're differentiating into neurons, nerve cells, basically. But again, interestingly, if you add this reagent called blebistatin, which is this cross, this is this line down here. Blebistatin is, again, like BDM, an agent that kills the activity of the stress fibers. Again, you've stopped the differentiation behavior. You've basically stopped the way that the cells are detecting their environment, and you're not getting any kind of differentiation. So it's something which is saying that mechanical cues are providing, it's these mechanical stresses that are telling the cell what to do, at least in these sorts of, experiments. And similarly, you can also create geometrical cues. So if you take the cell on a particular patch, you put a patch of fibronectin, which is a protein that the cells like to stick to, and that's how you control the cell shape. If you keep the cell area fixed, and if you put a square patch, they basically have equal probability of differentiation into adipocytes, fat cells, and osteoblasts, bone cells. But if you keep the area same, but you make them elongated, then they want to differentiate into bone cells. So these are the sorts of mechanical cues, geometrical cues that we are really interested in. Again, if you had added blebistatin to this experiment, this behavior would have gone away, basically. So this is the sort of overall thing that I want to sort of try to understand. I will not say that we'll be able to understand all these things, but maybe some aspects of these things is what's going to be the aim of this talk. Okay, so let's talk about starting from trying to develop this so-called constitutive model first. So in the very simplest case, let's see what would mechanical force, applied stresses or forces want to do. So one of the first sort of classical experiments that people try to predict is the following. You basically take a cell on a substrate and you cycle that substrate in one direction, two directions, etc. And I won't go into details about this, but this is just to motivate the mechanical model. And the application of this is sort of, that's what's happening inside an artery or something like that. That the cells on the endothelial cells on the inside of the cell wall are subjected to cyclic loading as your heart pumps blood. So what's interesting about these experiments is that if you put cyclic loading in this direction, the cells seem at 1 hertz, the cells seem to orient perpendicular to the direction of cyclic loading. That's the observation. So let's just start by using this to motivate the development of a constitutive model. So as I said to you, what we're doing is the stress fiber model. So now I'm going to spend the next 10 minutes or so maybe trying to sort of describe this constitutive model in very broad terms, because otherwise you're not going to get the rest of the talk, really. Okay, so to understand this, we'll do exactly what we would do typically in a materials thing. We'll understand the microstructure, and then we'll motivate a constitutive model from that microstructure in some sort of phenomenological sense, in this case at least. So you have a stress fiber. It looks like this. It comprises of lots of these functional units. Each functional unit looks like this, where this is the myosin filament and these are the actin filaments. And this functional unit, of course, has an internal state variable because I can either pull it. So this would be in the ground state. If it's stretched, it would look like this. And if it's sort of compressed, it would look like this. And there's some internal state variable that's describing what is the strain state of that particular functional unit, basically. And you have lots of these fibers. These fibers can be in different places inside the cell. They can be at different orientations. And so we think of a large cell when you think of some kind of spherical RV inside that cell with these stress fibers at various orientations. and then you describe the number, the number density, angular number density of these stress fibers. So at any given angle, you say that eta is the angular concentration of these stress fibers. It's basically telling me what is the concentration of these fibers at a given orientation at a given position inside the cell, where each of those fibers is what I just described before. Okay, so that's standard, and then you say to yourself, okay, these stress fibers need to form. They're forming from the soup of proteins that exists inside the cell. So you need to have some kind of kinetics for the formation and the dissociation of these stress fibers. And remember, at this point, we are doing something which is pretty standard, because these are proteins. They're going to follow Boltzmann statistics in some way or the other, basically. So we will use some kind of Boltzmann. We have an energy landscape for unbound proteins, these are all the proteins sitting inside the soup. This is the proteins when they form a stress fiber. And you have some kind of energy landscape that describes these two different states. You can also you have some activation barrier to go from one to the other. And you then use some kind of Boltzmann statistics to understand the kinetics as well as understand what is the equilibrium, of course, between this state and this state. So that's what we're going to do. But of course, where the phenomenology is going to come in at this point is to say that we need to have some kind of expressions for these different enthalpies, basically, because that's what's going to govern everything. So these are just some proposals, really, for these enthalpies. So in the unbound state, these are just free proteins. You'll say that the enthalpy is just in the absence of an activation signal, it's looking like this, but you always get some kind of activation signal. So in the presence of the activation signal, the enthalpy is modified in this manner. So for the unbound state, it's very simple. For the bound state, it's much more complicated, and it's a little bit more subtle. So this is the expression for the bound state of the protein, and I'll just spend 1 minute on this, basically. So in the bound state, this is the enthalpy of one particular functional unit. Of course, for all those functional units, you can just add up the enthalpies. So there's going to be an internal energy, which is going to be a function of some of that internal strain state that I described. And I've just put a quadratic form because I know nothing else at this point. But there's a second term here, which is this term here. This is what you would call the PV term of the enthalpy, basically. This is sigma F here is the stress that is being internally generated by these stress fibers. And this is what is going to, this stress here is tensile, and that's, of course, that's why I've put this minus sign here, is what's going to sort of change the, is going to change the sort of the energy landscape. Because this is the key point in biology, is that this sigma omega term, basically, the PV term, is typically on the order of the internal energy. And this term is going to be governed by the external mechanical equilibrium as we go on to discuss this, basically. And as you govern this term by the external mechanical equilibrium, that will completely change the structure of the stress fibers that will form, because it will change the enthalpy of these particular proteins. That's where, this is the coupling between the chemistry and the mechanics that's going to come as we go along. There's one more aspect to this whole thing, is that how many functional units would make a stress fiber. And that basically, I will again run through this a little bit quickly, is what's known experimentally is that these functional units, come what may, whatever being the external state, are typically on the order of 2 microns, basically. And so what you say is that if you come and pull the stress fiber, you extend it. Of course, initially the functional units will extend, but then the entire stress fiber will remodel so that one more functional unit comes back in here, so that all the functional units are again approximately at the correct length. So they are always going back to some resting initial state. Okay. Now you say, where does this term that we had, this sigma S, basically, this particular stress, what governs that stress? You need some kind of a stress-strain relationship or stress-strain rate relationship. And that also is well known. There's a lot of things that are well known about these stress fibers because they're muscles, right? There have been hundreds of experiments done from early 1910s, basically. Hill was doing experiments at University College in London in 1910 and 1917, etc. during World War I. I don't know why he was interested then in muscles, but he was doing these classical experiments, basically. And he found out that what we typically know of is that the stress-strain rate relationship, this is strain rate, for a muscle can be approximately approximated like this. But this is the critical part. It's basically saying that if the strain rate is 0, the stress that the muscle or the stress fiber can generate is very large. But as the strain rate becomes more contractile, then the stress decreases. And we know this. If you think about picking up weights, you can pick up a heavy weight very slowly, but you can pick up a light weight rather quickly. And that's what's being captured in that relationship here, basically. And so that gives me the stress-strain rate relationship. And this is extremely well characterized in the literature. There's also strain dependence, and the strain dependence comes from if you pull the stress fiber too much, there's nearly no overlap between these myosin filaments and the actin filaments, and so, of course, the stress will drop. If you get too much overlap, again, the stress will drop because you're getting interference of these filaments, and somewhere in the middle, you're getting a maximum stress. So this will be the stress-strain relationship. This is a stress-strain rate relationship, and so you can put that all together to get that sigma f. And now I'm going to say, okay, so these are all the constitutive parts. Now you have to come up with conservation laws. And so there are essentially only two conservation laws here. One is the conservation of proteins, the conservation of mass. You have a total number of proteins, you have an unbound number of proteins, and you have a bound number of proteins. That has to remain constant. So at any given point in the cell, you have a certain number of unbound proteins and a certain number of bound proteins. The bound proteins you need to calculate over all the stress fiber angles, and that's why I'm showing you these integrals. Now, the unbound proteins are free to diffuse in the cell. The bound proteins are not going to diffuse, basically. And so you have a conservation law basically saying that the integral of this quantity over the whole cell is conserved, and the evolution, the time evolution of this quantity at any given point is given by some kind of diffusive relationship of the unbound proteins. So I'm not writing the equations, but you can see that. So that's conservation law number 1. Conservation law number 2 is, of course, the obvious mechanical stress conservation law. You know what is the stress inside each stress fiber. You can then integrate that over the RV, over the spherical RV, and calculate what would be the stress tensor generated by those stress fibers. And that will give you the active stress that you get in the stress due to the stress fibers. There's also going to be some passive contribution. This passive contribution is basically saying that all the rest which I have ignored, saying the cell wall, all the other elements of the cytoskeleton, I'm dumping for the moment into the passive elasticity. You could develop a far more complicated model where you have all that sitting in there. But for the moment, I'm saying that there's only this active component due to stress fibers, there's a passive component, and the total stress is given, of course, by the sum of the active and the passive, and that will, of course, have to follow some kind of mechanical equilibrium relationship. Okay, so you do all that, and you come up with some prediction, and you can now say, okay, what will be the cyclic response? So I do a cyclic loading, just like in the experiments, on the substrate, and you first put it under static loading. So after the static phase, this is 6 hours of static loading, you basically get that the stress fibers are isotropic. There is no, that the stress fibers form in all directions with equal intensity. But then you put another 6 hours, so going to 12 hours of cyclic loading in this direction. And then the stress fibers this is the prediction, the stress fibers have now oriented preferentially perpendicular to the direction of loading, sort of consistent with these experiments. This is a deterministic prediction at the moment, okay? And so you'll say, why did this happen? The reason it happened is very simple, and the reason is the Hill relationship. The Hill relationship said that if I did contraction, I got a reduction in the stress, right? That's what we said, that if I'm having a contractile strain rate, I get a reduction in the stress. When I get a reduction in the stress, that enthalpy term of the bound stress fibers basically goes up. That destabilizes the stress fibers, and that means I get more dissociation in this direction compared to this direction because there's contractile strain rates in this direction, which means I'm getting a more preferential orientation perpendicular to the direction of straining. So this is the basic idea that is happening in this deterministic prediction. So I'm going to stop at this point and say this is the constitutive model, and I needed to explain the constitutive model to say, what are the elements here. But the main point, as you'll see from here, is that, look, when you did these experiments, every cell, again, was a bit different. Here we have got ourselves one deterministic prediction. So there's obviously something which is sort of unsatisfactory, if nothing else, basically, about this particular approach. So I'm going to now move on to saying that I've got myself this model. this model will be able to give me the energy of the system at any given state, etc. It will be there. And now we'll start trying to see that what can we do to understand this variability that is typically observed in these experiments. Okay? So that's what I'm going to talk about, a statistical mechanics description. So what is it that I'm going to do? I'm going to think of a single cell on an elastic substrate, and you'll see why I'm going to need to constrain myself to an elastic. Elastic, of course, includes rigid and all sorts of topologies, but not a viscoelastic substrate or something like that at the moment, okay? So I'm going to constrain myself to an elastic substrate, and of course, these cells do not exist in isolation. They'll die in isolation. They always sit inside a media. This media is basically a solution with lots of stuff inside it, essentially nutrients inside it. And so this media has all these nutrients, and of course, there's this external atmosphere. Now, the system that I'm going to be describing, and this is, of course, very important from a thermodynamics perspective, is the system that I'm going to be using is only the cell plus this substrate, not this media. Now, this is very important because, you see, if I had included my media into my system and only kept the external atmosphere being as the environment, then I would have got what we would typically think of a canonical system or a Gibbs ensemble, and I would be able to apply all the traditional statistical mechanics in terms of Gibbs, basically. But the problem is that my observable that I have is only the cell. I don't really care about what's happening inside this media, and in fact, in the experiments which run for many days, I come and input some more nutrients in it over some time, etc. So I really want this media to be be external to my system. But the problem when I have this media external to my system is that the interactions of my system now with its environment, which is this media, are not just thermal interactions like in a Gibbs system, thermal or mechanical interactions. This media and the cell are exchanging nutrients and all sorts of other complicated chemistry. And that's what's going to create some non-traditional statistical mechanics in this system, basically. So I've given you the whole story, but now I'll go into the details. So what I'm going to do is I'm going to funnily describe the system of only being the cell plus a substrate for the reasons that I've described. And then I'm going to somehow or the other try to use the second law of thermodynamics, which is entropy maximization, to try to understand the equilibrium of the system, even though it's going to be strongly interacting via nutrients with this media, basically. Okay, so how am I going to do that? And just to make sure that, this equilibrium is a strange concept, right? So the equilibrium I'm going to be discussing is from one day to a few days, basically. It's not going to be over, my lifetime, for example. It's not going to be 70 years or something like that. Okay, so that is the equilibrium that I'm discussing. So now to come up with the statistical mechanics description, we'll talk about two levels of microstates. Okay, so microstate in statistical mechanics is when every possible sort of variable inside the system is described. So one is an ensemble of molecular microstates. So these would be two different ensembles of molecular microstates. What does that mean? That in a molecular microstate is what a traditional statistical mechanics person would be describing. The molecular configuration is perfectly specified. So the ensemble of molecular microstates is that each molecular microstate has this very specific molecular configuration. You tell me the position, you tell me the velocity of every molecule inside this particular system. Okay, so that's the first level of ensembles. The second level of ensembles is what I'm going to be calling the ensemble of spread microstates. Now, the spread microstate is what you actually observe. So a spread microstate is basically telling me what is the connection between the cell and the substrate, for example. So if you give me the mapping of the cell membrane points onto the substrate, you give me the perfect shape and exactly how that shape was generated by mapping of the cell onto the substrate, that gives me what is a spread microstate. Okay, so that's it. So each in this particular experiment, in this particular sort of thing, each of these little fluctuations that you're seeing is one different spread microstate. Okay, so that's what you're seeing, and of course, every molecular microstate can only belong to one spread microstate. Okay, so now you have these two levels of microstates. And in a statistical mechanics sense, you'll define a macrostate. What is a macrostate? Is giving me the probability distribution of finding a particular microstate. Okay? So a molecular microstate is evolving on the orders of tens of seconds. because diffusion timescales are on the orders of tens of seconds, if not, maybe a minute or so. And that's what's basically telling me over what timescale is the molecular macrostate evolving. So the probability distribution of the molecular microstates are evolving on the orders of tens of seconds. The spread microstate on the other hand, when you're actually changing the shape of the cell, that occurs by processes like actin treadmilling, and I'm not going to go into those details, much slower timescale processes. And those microstates are changing on the orders of tens of minutes. Okay, so the spread macrostate is changing on the order of tens of minutes. And that means that I've got a separation of timescales. There's a temporal decomposition of the timescales between these molecular microstates and spread microstates, and I'm going to use that to look at the equilibrium system. Okay, so now let's go on and discuss something which is, entropy maximization, basically, in these in this scale, basically. So this is traditional stuff. You describe a Shannon entropy or a Gibbs entropy, and that's given by this quantity, which is basically the sum over the probabilities of a particular molecular microstate. But a molecular microstate can be decomposed into a joint probability between the molecular microstate and the spread microstate in this manner. So I'll first sum over all the molecular microstates in a particular spread microstate, and then sum over all the spread microstates. And I'll just start skipping over some of this algebra. So you can now start using Bayes' theorem, and you can write conditional probabilities. It's going to make life simple because I'll be able to do some temporal decomposition. And when I do that, when I use Bayes' theorem, basically I get two entropies. I get a spread entropy, which is given by this term, and the typical molecular entropy. And now I'm going to do equilibrium of this particular expression. Now the first thing is I'm going to do equilibrium on tens of seconds. When I'm doing equilibrium on tens of seconds, the spread microstate is not changing. So the variation of the probability distribution of the spread microstate is not changing. But this particular conditional probability of a molecular microstate is changing, and then I'm going to try to maximize that particular entropy, and it's going to give me that value. But this is not unconditional maximization. This is Gibbs, basically. You're going to maximize it subject to some enthalpy constraint, and of course the sum of the probabilities is 1. And this is what Gibbs did. He did this. He enforced those constraints by Lagrange multiplier. The Lagrange multiplier in this case turns out to be essentially the thermodynamic temperature, and you get the expression, the standard expression for the Gibbs free energy. And then, of course, the equilibrium will be when the Gibbs free energy, the variation in the Gibbs free energy is equal to 0. So that's what you get from when you're saying that the spread macrostate is not changing, and you're getting only changes in the molecular microstate. You'll get the standard idea. The standard idea you'll get is that the equilibrium of the system is at the minimum value of the Gibbs free energy, basically. And the Gibbs free energy would minimize, of course, as I'll say. So now you're talking about the equilibrium of the spread microstate. So it's a standard boundary value problem. You'll get minimization of the Gibbs free energy. So you've defined the spread microstate, and then you now need some kind of an energy model. The energy model is what I had been describing all this time earlier, basically. You do that, and then over that time scale, you find mechanical equilibrium and chemical equilibrium. Those chemical equilibrium equations that I spoke about, diffusion, kinetics, etc., and the mechanical equilibrium equation. And you get the minimum Gibbs free energy solution, which means that I get this particular value. So this is, you do this in a standard mechanics way, and you solve this multi-physics problem, and you get yourself the minimum Gibbs free energy solution. Okay, you've got that, but you've got that for one particular spread microstate, basically, right? For one particular spread microstate, it's telling me this is my minimum Gibbs free energy solution. Now, what happens over many hours? If I'm saying that was equilibrium over tens of seconds, basically. Now I want to look at equilibrium over tens of hours, basically, or at least 1 day or 2 days at least. So then I have to add the constraints from those lower timescales into the expression for the Gibbs free energy, into the expression for entropy of the total entropy expression. And now I'm going to again have to look at maximization of this entropy, but now the spread macrostate is changing, basically. So this term is not equal to 0. So I will have to maximize this subject to this not being equal to 0. There's the usual constraint, the temperature constraint that Gibbs had, carries through. It was there at the small time scale. That temperature constraint is carrying through. So this particular temperature constraint has carried through. Now the question is, what else has to be done? So if I had said that only this particular temperature constraint exists, then I would have just ended up saying that there's going to be one deterministic free energy, minimum free energy solution, and I would get the Gibbs sort of idea, which is what we have typically been using, holding true. But at this point, because I have an option of adding another constraint, if there's a physics, basically, to add another constraint, basically. So the question is, what additional constraints do I add to this maximization, if any, basically? And I'm going to just slow down a minute here, because this is the critical aspect. So what are the other constraints? And I'm going to invoke a concept called cellular homeostasis. This is not my concept. This was first proposed in 1865 or something like that. And what is cellular homeostasis? We all know it, right? You approximately can maintain your body temperature at some fixed value. Hodgkin-Huxley, won the Nobel Prize for ionic pumps and things like that, when they said that irrespective of what's happening to the environment, the cells have mechanisms in them by which they can maintain the ion concentration inside them at some specific value, etc. So what cellular homeostasis is a whole bunch of very complex biochemical processes. I'm not going to say that I know any of them, basically, but what I do know that they exist and they maintain the various molecular species within the cell at a specific average number, not fixed at a particular number, but over a long time scale, i.e. a few hours, etc. The average number of proteins of any particular kind of species inside the cell will have a fixed average number. And that constraint was not there. If I just continued to do the Gibbs ensemble, this cellular homeostasis constraint would not have existed. So I'm now going to add this constraint. But how do I actually add it? It's a fine statement to make like this, but what does it physically mean? Well, I'm going to show you that what this physical constraint means is that it imposes a constraint on the average Gibbs energy. It's a very strange thing to say, that there's an average Gibbs free energy, because you always think of some kind of minimum Gibbs free energy. I'm going to be saying that there's an average Gibbs free energy that will be constrained at some value. So how does this happen? Okay. Oh. So let's think of a freestanding cell, i.e. a cell in free space, okay? A cell in suspension, basically, which is E sub is equal to 0. Now, this is a unique boundary value problem that I would notice, as I would call it, and it would say that the tractions over the entire cell are equal to 0. It's freestanding, basically. And that would admit only one single solution. It's a boundary value problem. There's going to be a unique solution to this one boundary value problem, and it will give me a Gibbs free energy if I did that boundary value problem solution, and I'll call that Gibbs free energy G naught. And of course, that would be written as the sum of the chemical potentials multiplied by the number of each species over all particular species. Okay, so that gives me that value for a freestanding cell. Now I take a cell on an isolated substrate, take the substrate isolated from the cell. Of course, the substrate has some energy because it's just sitting there isolated. Now I bring the cell into contact with the substrate, okay? So I bring the two into contact. What will happen, and I'm going to say that it is in some spread microstate J, the cell, and so the system is in some spread state J. You could write that free energy as G naught, which is that term here, plus, of course, these would be the two terms from the isolated case, plus some interaction term, because there's going to be some interaction that's occurring between the cell and the substrate. And then the average would be over over the entire ensemble would be given by this term here. Now, these are obviously simple terms. It's this term that is a bit of a crazy term. So let's think about how do I get hold of this term, because I know nothing about this term. So first thing I'll say is let's get hold of this delta G term, right? What does this delta G term look like? So if you go back to the Gibbs-Duhem identity, in a constant temperature, constant pressure situation, the Gibbs-Duhem identity will tell me that delta G is given by this, where this is the sum over all cell species, and this is the sum over all substrate species. Now, what's interesting about all of this is that the substrate species, this particular term, because it's an elastic substrate, that will be equal to 0. And I'm seeing I'm running a bit out of time. So that will basically tell me this reduces to an expression which looks something like this, basically. And this term, will basically tell me that I get this, and it's basically telling me that this term is exactly equal to what it is for a freestanding cell, because the number of proteins this is cellular homeostasis. The number of proteins average is independent of the cell substrate, basically. And so that's what I'm basically saying, that this average G is basically just the G of the substrate plus the G of the freestanding cell. Okay, so I do this. I put it into, I do this constraint maximization again, and I get the probability distribution of a spread microstate to look like this. And I don't know, I have a Lagrange multiplier beta that I don't know how to deal with, but I have to solve this funny equation to get hold of my Lagrange multiplier beta, which is the conjugate to my spread entropy. So now I've got myself a new type of temperature, which is different from thermodynamic temperature here. And of course, now I'm saying that the system is going to be in equilibrium, not at the minimum Gibbs free energy, but at some new potential. And this is what I'm calling the spread potential, this being the usual partition function. And basically, it's telling me that I'm getting, M, the spread potential, is exactly analogous to the Helmholtz free energy, basically, for a canonical ensemble. So I've got myself this expression, and you think, okay, you've done it all analytically. The problem I have here is that I've got the probability of a spread microstate, but I have no idea what is the spread entropy. And to calculate that spread entropy, how do I do that? I use Markov chain Monte Carlo methods, basically. So in a standard Markov chain Monte Carlo method, you'd use a Metropolis algorithm. For that, you'd need to know the temperature. In my case, beta, I don't know the temperature, so I use other algorithms called nested sampling and some other more complex Bayesian schemes. We would be running very, very large number of calculations, tens of millions of FE calculations, basically, now to do the sampling over all particular spread microstates. At each of these steps, we'd have to solve that entire FE problem of the coupled biochemistry of the cell and the substrate. And we solve that by a mixed finite element boundary element method. The substrate is done by a boundary element method. The cell is done by a finite element method. So you do that, and this would give you some examples. So for example, a cell on a rigid substrate, these would be all the sampled microstates. Remember, don't think of this as a time-lapse movie, okay? These are just samples of the microstates. If you put the cells on a particular substrate, this is what it would look on a constrained patch. This is what it would look like. If I put the cell on a very large patch, this is what it would look like, So, for example, the cell will also become mobile at some point. So it will start to move, etc. So motility and things like that would also be coming out from these kinds of analyses. Okay, so what do you actually get? So if you have done all that, and you basically will find that what is the probability of observing the system in a particular free energy. So if I put the cell on a 70 kilopascal substrate, this is what I would say would be the energy distributions of the cells. If I put it on a softer substrate, 5 kilopascals, that would what it would look like. And if I put it on a 2 kilopascal substrate, these are what would be the energy distributions that would look like. So what you're firstly seeing is that as I go to softer substrates, the probability distributions are narrowing down, basically. And of course, remember that at each particular point, so for example, at this mode of this 70 kilopascal thing, you get hundreds and thousands of these different configurations. But this is just telling you that all these have the same energy, but they're all different shapes and sizes, basically. So first thing you're seeing is this narrowing of the distribution as you go to softer substrates. If you say, but that's no use because that's energy, I can't measure energy. What about the area distributions? You take those same calculations and you look at the area distributions. This is how the area would be distributed as a function of the substrate. This would be the probability distribution of the area on a 70 kilopascal substrate. This is what it would look like on a 5 kilopascal substrate, and this is what it would look like on a 2 kilopascal substrate. So again, what you're seeing is that the variation is decreasing with decreasing substrate stiffness. But in this case, of course, the mean value is also becoming smaller as you decrease the substrate stiffness. And this is consistent with the experiments of Dennis Disher. So if you compare experiments and predictions, they are pretty much in line with each other. And it's also showing you how the variations are decreasing with decreasing substrate stiffness, as observed in these experiments, where you saw those big variations were decreasing with decreasing substrate stiffness. We get the same thing with aspect ratio. We can do exactly what's done in the experiments for different shapes. We can fit ellipses to these cells. This is what the experimentalists would do. And we can then plot probability distributions of aspect ratio. And in general, we see exactly the same thing, sort of. We see here, you see in the experiments, you see that the peak aspect ratio decreases with the decreasing substrate stiffness. And we see that, not to the same extent, I must admit, but we approximately see that happening here as well. I'm thinking I'm running out of time. We can take the same thing forward into stem cell differentiation as well. These are experiments done by a colleague of mine, Chris Chen, and we've worked with him for many years, and he showed how stem cell differentiation depends upon the area of the cell. And again, if you add BDM, then you basically, or some other myosin inhibitor, you basically get no differentiation. And if you do the same, I won't go into the details, but you can extend this analysis to start getting into stem cell differentiation as well. I'll end the talk. I wanted to just spend 1 or 2 minutes by just sort of summarizing what I wanted to say. The main thing is that we've got ourselves a statistical mechanics framework to describe these kinds of variability observed in vitro experiments. The main thing is the following, is that unlike in a system in a thermal bath, in a thermonutrient bath, what you're getting is a system that has a fluctuating Gibbs free energy. It is not a system where basically this Gibbs free energy is constantly decreasing and you basically would get equilibrium at the minimum Gibbs free energy. And you know this, right? You and I, we all would be dead. The dead state is the minimum Gibbs free energy state, basically. And so what we have basically said is that there's something else happening, and cellular homeostasis was used, basically, to understand what is this new equilibrium, basically, that you're going to be getting. So what you're saying, basically, here is that the equilibrium is occurring not at the minimum Gibbs free energy, but at this new type of potential, which I've just called the spread potential in this particular case. And of course, we've used this model to understand, mechanical, topological, and all sorts of chemical cues for how cells detect this chemical cues, etc. And what comes out from this analysis is cell contractility, the stress fiber contractility, spreading, motility, are all coupled phenomena. You don't think of them separately, and they are set by the fluctuations of the cell on this hyper-energy, Gibbs-free energy hyperspace. Not an energy hypersurface, but a Gibbs-free energy hypersurface, basically. That's what's happening in this case. And the final point I'll say is that you can extend these ideas, of course, to stem cell differentiation, but this is the main point, is the statistical mechanics framework is independent of the underlying energy model. I gave you an energy model to start off with. That was my example. That could have all sorts of problems with it. It has all sorts of simplifications in it. And that is a constitutive model. We can keep modifying, improving that constitutive model, but you analyze it you use that constitutive model to analyze sort of what's happening within this framework. And that's typically what's done in normal statistical mechanics as well. The different molecular people will come up with 50 or 1,000 different potentials, but they will use the Boltzmann statistical mechanics framework to analyze it. And I'm not saying that whether this is a correct statistical mechanics framework or not, that may be left for argument. But something like this should be done to try to understand this inherent fluctuations, because this is not just experimental error. This is actually physical in the system, and that needs to be sort of understood in some way or the other. Thank you very much.
Thank you very much for being punctual on time. We have time for a couple of questions from the audience, please. Anna, please.
Substrate, yes.
And then you took away some term in
Correct. So the point is that because it's an elastic substrate, the number of species, if you come and deform an elastic substrate, you only get a change in the chemical potential of the species, and the species themselves undergo no transformation. That is the nature of an elastic material, basically. And that is what I required to get that constraint. And if you told me that you had a viscoelastic substrate or something like that, then I would not be able to make that assumption, basically. Now, we have been thinking about how to relax it, but I haven't thought about it completely.
And then you mentioned always that it's in vitro experiment.
Correct.
And there is nothing done in vivo.
So people, of course, do in vivo experiments. The problem with in vivo experiments is the following, of course, they're real. You cannot deny that. But to control variables in an in vivo experiment is extremely difficult, basically. For example, here we come and add BDM, or we control the shape of the cell, the size. These sorts of controlled experiments are next to impossible to be done in an in vivo setting. So what you typically are trying to do here is that you're trying to recreate some kind of in vivo environment. That is one thing from a medical perspective. But from an engineering perspective, I think those in vitro experiments are even more exciting. Why is it that I'm interested in all this? Is we want to grow an organ outside the body, basically. That's the final goal. I'm not saying we are there, but that would be the final goal. And that is why we are trying to design these kinds of topological environments, stiffness environments, to see, how can you control stem cell differentiation, et cetera, in vitro in terms of trying to grow this organ. I will emphasize, nobody's anywhere near there, but that is the aim, basically. So there is a good reason, engineering reason, to think about actually designing good in vitro experiments as well.
And for one more question, Alessio, a little bit.
Thank you very much. I wanted to ask you about the eventual stresses that you could have from the fluid.
Yes. So, the framework, of course, it will, of course, increase the if you wanted to now have fluid flow, for example, in addition to something else, that's going to increase the computational cost of computing the energy of a microstate significantly, but in principle, it's possible. I must say that, for example, these calculations that I showed, typically required, 1,000 cores running for a week to do these levels of calculations. This would be a very significant, enhancement if you had to include fluid flow. And, even before that, I would want to go to 3D. These are quasi-2D kind of experiment simulations. So, but all of this, the real thing is that I would say 5 years ago, even this level of computation would not have been possible. These highly parallelized Bayesian algorithms that we use to do these entropy calculations. But they have just about become possible. So, maybe possible, I'm not sure.
You factorize the entropy of the system as a condition that the average of the entropy.
Average of the free energy. So in the first time scale, it is average of the enthalpy. In the second time scale, it's average of the free energy.
No, just so that the difference also include the standard deviation of these quantities as a condition.
So,
also, not only the average,
yeah.
also.
So you could. The reason I have done it on the average is I tried to go through that derivation saying that there's a physical reason to, when you're doing entropy maximization, you can constrain it to it any way you want. You need to have some physical reason to do it. And I think that I have a physical reason to constrain the average, basically. If I could think of a physical reason to constrain the standard deviation, I would do it. Yeah.
Can you see other hands away? Well, one, Mr. Marino, please.
In the example that you show about the anterior segment,
Sorry, can I didn't.
in the example that you show about the anterior segment, you
Yeah.
integrate everything on the fluid flow.
Oh, no, sorry. Okay, so I should clarify what I was. So in the arterial example, there are two things which are happening. One is fluid flow, and the other is just this kind of cyclic loading, cyclic expansion of the artery. In the experiments that I was describing, the fluid flow, these were in vitro experiments. So they were put cells on a substrate, and they cycled only the substrate with no fluid flow on it. So in those experiments. And so hence in the model, we only included the cyclic loading. Now, people have definitely done experiments where they have done cyclic loading plus fluid flow as well.